Good evening, everyone. My name is Magda Gerik, and I'm from Department of Engineering. It is a pleasure to be here tonight, and I'm delighted to share with you some of the results from my PhD project. My PhD project was very much methods and protocols development oriented, but the main idea behind it was to create a platform that would allow us to build a model of a brain tumor, a very specific brain tumor that I will talk about in a second. I know that the title might sound a bit enigmatic, but hopefully I can explain to you what I had in mind when I was doing it. So going back to the aim of why I actually decided to create a 3D model to study brain tumor progression is that brain tumors are very tricky cancers to treat. They are actually considered as one of the 4 hard-to-treat cancers at the moment. And the most aggressive form of primary brain tumor, and unfortunately also the one that is most commonly identified in patients is glioblastoma. Glioblastoma is characterized by very high resistance to therapies. It is a very infiltrative tumor, and that means that cells from the tumor mass can migrate away from the tumor mass and quite literally hide in vascular niches, and because of that, the tumor can then recur after certain time, and that also leads to tumor progression. Another aspect of glioblastomas is the fact that they have a very specialized type of cells within the tumor mass, but also within the cells that migrate away of the tumor mass and infiltrate the healthy parenchyma, and those cells are stem-like cells. Unfortunately, the standard treatment of care, which at the moment includes surgical resection followed by chemo and radiation therapy, those therapies do not take into account the dynamic involvement of tumor microenvironment, which is usually the cancer cells. They try to recruit the elements of healthy brain to help the cells to actually progress the tumor. So the 2 aspects of tumor microenvironment, which I was trying to recreate in my 3D environment, were extracellular matrix, so all of those components, like for example collagen, that allow cells to move in the space in the brain, and also blood vessels. And in brain, we know that it is a blood-brain barrier, so a very tight and very strong vessels that do not let, for example, chemotherapeutics to cross it, but the brain cancer cells unfortunately found a way to even recruit those vessels for their benefit. One of the aspects of glioblastoma is the fact that they can grow and progress without angiogenesis. Angiogenesis is a mechanism where tumors they produce new blood vessels. It is a formation of new blood vessels, and angiogenesis has been a very established mechanism for tumor progression, not just brain tumor, but pretty much every cancer in the body that you can find. However, brain tumors, they have found a way around it. So instead of forming new blood vessels straight away, they switch on different mechanisms. And one of the mechanisms is vessel corroption. It is a mechanism where cancer cells, they migrate towards a healthy vessel. They can then wrap around that vessel. They can get nutrients from it. They can migrate it along it. And that way they can actually recruit it to then start a new tumor, or they kind of incorporate it in existing tumor mass. So it is a big challenge to overcome, and it is a very complicated mechanism that we didn't really know how to study, and the reason for that is that the vessel corruption mechanism has been hypothesized from looking at slices of brains with brain tumors resected from animals, but as you can imagine, this is only an endpoint analysis, so you look at it and you see that, oh, this is something new that I haven't seen before. However, there is no way of actually tracking it in real time, so we don't really know how it looks like, whether the cells cooperate with each other first and then migrate towards the vessel, or whether it's just one cell going towards the vessel. So for that reason, I decided to build a 3D microenvironment, which would include an artificial brain microvessel, as here, and cells, brain cancer cells, including those stem cells that I mentioned, would be embedded also in 3D microenvironment. So this is also a time to mention that at the moment, a lot of studies in biology, they happen in 2D, where the cells are cultured in flat environment, and as you know, our cells in the body do not really work that way. Therefore, it is very important to try to recreate the physiological scenario here. So the device that I designed, optimized, and validated looks like this. It is a very tiny device, as you can see, but at the same time, this is the very similar scale of what would be happening in situation of a brain tumor. And those little devices, they're called microfluidic devices, and microfluidics as a set of techniques, it has been around for nearly 30 years at the moment. And engineering-wise, those devices are great. They are very advanced. They're just lovely things that you can study using them. However, the translation from the engineering world to the biology world has not been that clear. And the reason for that is because biologists, they don't only want to see visually what's happening, they also want to see what's happening on RNA level, on protein level, and for that reason, those devices have not been utilized in a specific way. It is, as you can see, it is very small, therefore the volumes are very small, and the number of cells is very small as well. However, it is tricky to retrieve the cells from the device, but it is not impossible. So that was one of the part of my work, that I managed to track the cells in real time in the devices and then actually retrieve the cells from it so that we can check what is happening on the genetic level. And everything was designed so it would be very easy to incorporate in day-to-day biological assays. So that's why it looks very not impressive, but this is something that biologists can use, and that was that was the aim of my project. Results-wise, because brain tumors, and especially glioblastomas, they're very heterogenic. They really vary from patient to patient and also within the tumor itself. Therefore, it's very important to me to check that in my microfluidic device, many patient-derived cells could survive in the cells. This is so-called life and death assay, where we check whether the cells can actually grow and progress in the environment that we are subjecting them to. And it was quite a vital point to see that it was something that we could potentially in the future use for a patient-specific therapy finding. And it was also important to make sure that the cells inside the 3D environment, they had still their stemness signature. Therefore, those cells that are those, I'm going to say, scariest cells, in a way that they are very resistant to any therapies. And this is what we wanted to check. We wanted to check how those cells crosstalk with the endothelial cells. Oh, sorry. So the other parts of the device, so we have the cells embedded in the extracellular matrix, is the artificial microvessel that we create. So I decided to create vessels from brain microvascular cells, from lung microvascular cells, and also from umbilical cord endothelial cells. And the reason for that is that for years, people have been studying the interactions between brain cancer cells and endothelial cancer cells using cells derived from umbilical cord. And those are the cells that are very prone to angiogenesis. Therefore, it got concluded that, that brain tumors are very angiogenetic, which is not really the case because they might acquire many different mechanisms. That's why I wanted to make sure that the interactions that I was studying in my devices were actually tissue specific, hence 3 microvessels. The next step was combining both a microvessel here and the cells from brain tumors and checking whether they can survive in this device. So in engineering, and also not just in engineering, but it is very important to make sure that the materials that you are using and the environment that you are subjecting the cells to, this is the environment that the cells can actually prosper in a way that they would in physiological scenario. So we checked whether the cells were surviving, but the most important part is that we actually saw the differences between different patient-derived cells and how they influence different endothelial cells. So this is something that we need to take into account, that every type of cancer cells derived from patients and every subpopulation of the cells can influence endothelial cells in a different way. And this is a very important fact in terms of therapy, because unfortunately, anti-angiogenic therapies have not been working well in brain tumors. And the reason for that is that, first of all, there are different mechanisms that brain tumors can acquire, and the second is that unfortunately brain tumors can develop resistance to anti-angiogenic therapies and then switch to a different mechanism. So those kind of 3D devices can really help us see what the dynamics between the interactions are. And lastly, using the device, you can actually track the cells and what they are doing in time frames. So you can click on literally every cell and then see where the cell is going. And using that, we can see if cells with a specific signature are more prone to interact with a microvessel than other cells. We can see what their speed is, what their velocity is, whether they're actually polarized towards the microvessel. So all of those things, although being basic science, we believe that they can help with creating and actually testing new therapies for neovascularization, because at the moment, as I said, we are not only talking about angiogenesis, but about neovascularization in general, about providing the brain tumors with nutrients that they can get through many different mechanisms. And to summarize, this is a very short summary, but it kind of like just says what I was trying to achieve, was to recapitulate an in vivo-like condition, so really trying to recreate the physiology of what is happening at the micro scale, but also to find questions to some fundamental some answers to fundamental questions. And that's why I was really trying to optimize the device so that we can get analysis on many different aspects. So not just tracking the cells and seeing where they migrate towards, but also seeing what is happening to them at the genetic level. And I would like to thank my group, especially my supervisor, Dr. Sherry Huang. So that was my PhD project. I just finished. I had my viva 2 weeks ago, so I'm no longer working on this project. However, I have a new project that also concerns brain tumors. However, at the moment, I'm working on implantable drug delivery devices. It is also at the Department of Engineering, but I moved to electrical engineering, and I'm working in bioelectronics lab. If anyone would like to get any more information, I'm really happy to talk about my new project as well. But thank you.